Hi, thank you. I'm glad I got the link. Okay, so do I share my screen? Is that the right way?
Yep, go ahead. So I will briefly introduce you. So our speaker is Layla Parast. She's a senior statistician at the RAND Corporation and co-director of the Center for Causal Inference. Feel free to take it away whenever you're ready.
Okay, let me figure out how to share. Mm, Oh, start share. That looks promising. Okay, do you see something?
Yes, looks good.
Do full screen. Okay, all right. Thank you guys. Sorry for being a little late. Okay, so thank you so much for the introduction and for inviting me here today virtually. So today I'll be talking about assessing the value of surrogate markers measured with error. This work is supported by a grant from NIDDK and is joint work with Tanya Garcia at UNC, Ray Carroll at Texas A&M, and Ross Prentice at the Hutch. So first, just an outline of the talk. I'll go through some motivation and background behind why I'm interested in surrogate markers and where they might be useful. I'll talk about some methods to assess the value of a surrogate marker, both in a parametric framework and a non-parametric framework, and then propose some methods to correct for measurement error within those frameworks. And then I'll apply the proposed procedures to data from the diabetes prevention program, which was a randomized clinical trial, and close with some remarks and limitations and a little bit about other work in this area. So, as I'm sure all of you know, many clinical trials investigating the effectiveness of an intervention or treatment often require long-term follow-up of patients. In those kinds of settings, the availability of a surrogate marker could potentially allow researchers to make conclusions about the treatment effect with less required follow-up time or less cost. So this is kind of the promise of surrogate markers. If we can find them and validate them, we can use them to replace the primary outcome. And if it's something that can be measured earlier than the primary outcome, then we could have less follow-up time. But even if it's something that has to be measured at the same time, but if it's less costly or less invasive, can still be a helpful replacement. So these are just a few examples. So in the first example, the primary outcome of interest is a diabetes diagnosis. And so depending on the patient population, you may have to wait a very long time in order to observe enough diabetes diagnoses to have sufficient power to test for treatment effect. So some potential surrogate markers of interest might be hemoglobin A1C or fasting plasma glucose or BMI. Another area of work that often requires long follow-up time is dementia. So here the primary outcome would be dementia onset, and some potential surrogate markers would be a brain image or cognitive score of some kind. And then lastly, a lot of the earlier work, and still work now really, on surrogate markers was in the AIDS research area. So here the primary outcome is disease progression or death, and the surrogate marker is CD4 count. So I've been kind of throwing around the words or the phrase surrogate marker. So one definition of a surrogate marker is a physical measurement, such as a biomarker, a clinical measurement, or a psychological test that can be used in therapeutic trials as a substitute for a clinically meaningful endpoint that's a direct measure of how a patient feels, functions, or survives, and is expected to predict the effect of the therapy. So while that's kind of a nice definition, it's not really clear what that means statistically. In 1989, Ross Prentice published this like landmark paper that basically every paper on surrogate markers since then references this paper, where he proposed a criterion for a valid surrogate marker, where he required that a test for treatment effect on the primary outcome also be a valid test for the treatment effect on the primary outcome of interest. Sorry, I don't know if the test for treatment effect on the surrogate marker, also a valid test for the treatment effect on the primary outcome. One of the operational criteria he proposed was that the surrogate should be able to capture the dependence of the primary outcome on treatment. So people have kind of interpreted this as, a surrogate marker, like a good surrogate marker should capture a large proportion, preferably all, of the true treatment effect on the primary outcome. And so based on this, there's been a lot of work on this quantity of the proportion of the treatment effect that can be explained by the surrogate marker. That being said, there are many quantities and criteria that have been proposed to assess the value of a surrogate marker. So this proportion of treatment effect explained by the surrogate marker is just one of many. There's my understanding that there's not really any agreement on a single best method that should be used. So in a meta-analytic framework, there have been methods and measures proposed, so where you have multiple studies instead of just one study to evaluate the relationship between the surrogate marker and the primary outcome. And a lot of work much more recently on this topic in the area of causal inference. So while there are many methods available to assess the value of a surrogate marker, most of them tend to assume that the surrogate is not measured with error. And then on the flip side, while there's a lot of methods to correct for measurement error, there's little methodological work that's like at the intersection of these two. So what to do when you're trying to evaluate a surrogate marker and you have measurement error. So in this talk, I am going to focus on the quantity that I mentioned, the proportion of the treatment effect on the primary outcome that's explained by the treatment effect on the surrogate marker. And I'm going to denote that as R because it'll be a ratio. And again, this is, by no means the best quantity or the only quantity out there. It's the one I have tended to focus on, and I like it because it's intuitively appealing, and it's easy to interpret, and it's frequently used in practice. So, if you can explain to a clinician that this surrogate captures, 90% of the treatment effect, obviously that's very easy to interpret. And there are, I think there are certainly other methods that could be used, but this is just one that I'm focused on here and focused on how you correct for measurement error when there's a surrogate and you want to obtain this quantity. So my goals are to illustrate the bias when there's measurement error in estimating this R quantity and to propose methods to correct for this bias in both a parametric framework and a non-parametric framework. So you might be wondering why I'm talking about both a parametric and a non-parametric framework. If you asked me, which one you should use in practice, I'm going to say the non-parametric approach because others, including myself, have shown how things can go wrong, and I'll show them here when you use the parametric approach and your models are misspecified. But there are some settings where the researcher really does believe that the models you have to specify truly hold. And so I think it's important to offer a solution for how to correct for measurement error within that parametric framework, if that's the method that someone chooses to use. But I do lean very heavily towards the non-parametric approach, and I will show you why. So I'll introduce a little bit of notation. G is the treatment indicator, and I'm going to assume throughout that I have random assignment. Y is the primary outcome, and S is the surrogate marker, which is either measured earlier or at the same time as the primary outcome, but with less cost. And my aim is to evaluate the utility of the potential surrogate marker S by estimating that proportion of treatment effect quantity. So in the presence of measurement error, I don't observe S, of course. I observe a mismeasured S, which I'm denoting as W. And so W is S plus U, where the mean of U is 0 and the variance is some sigma U squared. I'm going to assume that sigma U squared is known, and I'll talk about that a little bit more later. So my observed data include the primary outcome, the mismeasured surrogate, and the treatment indicator. So the parametric approach, or one parametric approach here, is to simply define, specify two regression models. So this is very straightforward. So you have one regression where your only predictor in the model is treatment. You have a second regression model where you kind of throw in the surrogate marker. And so then your proportion explained is 1 minus the ratio of these two coefficients for treatment. So you can imagine if you fit that first model and your beta 1 is, large and significant, and then you fit the second model where you add in the surrogate, and now your beta 1 star is, something very small, then beta 1 star over beta 1 is going to be something small. So 1 minus that is close to 1. So the surrogate is, capturing a large proportion of that treatment effect. On the flip side, if you fit that second model and beta 1 star is basically the same as beta 1, then 1 minus that ratio will be close to 0, and the surrogate is really not capturing anything. So the estimation of R is obviously straightforward. You just fit those two models and get your estimate for R. When you don't have S, and instead you have your mismeasured W, then estimation in that second regression model obviously is affected. And so this is what the bias looks like in that, using that parametric approach. So this is a simulation where those two models do hold, and the proportion explained is about 0.5. So when there's no error, estimation works very nicely. And when there is error and you ignore it, you see pretty substantial bias. So because the way to estimate R just involves those linear regression models, you can just write out exactly what that attenuation bias term is. So you're not really estimating, you're not getting an estimate of beta 1 star, but beta 1 star plus this attenuation bias term. And similarly for R, you can just write out in closed form what it is that bias term that you're going to see. So based on that, you can just do some algebra and flip it around and get a disattenuated estimator. So plugging in, all of the pieces you need, you take the beta 1 star that you estimate when you use the W, subtract off this attenuation bias term, and get a new disattenuated estimator. So for this disattenuated estimator, we show the consistency and asymptotic normality and derive variance estimates, closed form variance estimates. Of course, we always want variance estimates for our estimators. But here in particular, it's important because in terms of decision-making, some people have argued that you should use the confidence interval for R to make a decision about whether something is, explaining a substantial amount of the treatment effect. So it's been proposed that, you say something is a kind of good surrogate if the lower bound of the confidence interval for R is greater than 0.75 or 0.5. So having a way to estimate that confidence interval is especially important here. So a second way to correct for measurement error within this framework that we propose is using the SIMEX estimator. So SIMEX stands for simulation extrapolation. I had never used this estimation approach before trying to work on this problem, but it's a really neat idea, and I really like it. So basically, the idea is you generate additional measurement error, so as quantified by this 1 plus lambda times your measurement error. So you take your W's that are already mismeasured and you add like a little bit more measurement error. And you do this across a grid for lambda. And you also do it for a certain number of iterations. So here I chose 50 iterations. And so for each iteration and each lambda value, you get the estimate, whatever you care about. So here it's fitting that second regression model and getting the beta 1 star that you would get now with this like more mismeasured W. And so for each lambda value, you do that 50 times, and then you take the average over the 50 times. And now you have information about how beta 1 star kind of behaves as you have more and more measurement error. So this is the simulation part of it. And the extrapolation part is then you extrapolate back to a setting where there's no measurement error. So where lambda equals -1, because if we go back to that first piece in blue, if lambda equals -1, that's a setting where there's no measurement error. And you have to choose some extrapolation function here. So I will show you throughout what it looks like when you use quadratic extrapolation and nonlinear extrapolation. So let me show what that looks like. So this is just for one of my simulations. So again, for each lambda, I have lambda on the x-axis and I have r on the y-axis. R, of course, is just a function of that beta 1 star and beta 1. So for each lambda, I'm doing those 50 iterations, learning how beta 1 star and in turn r behaves for that lambda and plotting it. And then I just connect all of those lines, and that's what all of those dots, and that's what the black line is. And then I extrapolate back to lambda equals -1. The black dot that's shown there on the left is the naive estimator. The red dot is the truth. And then the green dot is showing the estimate that you get when you use quadratic extrapolation. And the blue dot is showing the estimate you get when you use nonlinear extrapolation. So for this one iteration of my simulation, the quadratic extrapolation doesn't work that well, although it's still better than the naive estimator. And the nonlinear extrapolation is pretty close. And these are just a few more iterations. So the top left is the one I already showed you, and the others are just other iterations. So showing you that, you know, sometimes it's very close, sometimes it's a little far off. So similarly for the SIMEX estimator, we showed consistency and asymptotic normality and derived closed form variance estimates, which were non-trivial. And we were particularly kind of happy to get the some closed form estimates for the variance that worked well because you know, of course, it would kind of be easy to just do like some bootstrapping or resampling, but with the SIMEX estimator, because you're already like doing a simulation within there, to do the simulation and then do bootstrapping would be like very computationally intensive. So it's nice not to have to do that. The asymptotics for the SIMEX estimator do rely on the extrapolation approximation. So when I say consistency, I can really only say it's approximately consistent. But when the extrapolation function isn't correct, the asymptotic bias for the SIMEX estimator is still substantially smaller than the naive estimator. And that's not something I showed, but it's something that Ray and some of the earlier SIMEX papers have shown. Okay, so these are some simulation results. So the first two plots you already saw. The first one is when there's no error and things work perfectly fine. When you use the naive estimator, you have bias. The disattenuated estimator is working well here. The SIMEX estimator with the quadratic extrapolation is, I mean, it's certainly reducing bias, but it's not as good as the nonlinear extrapolation. So that's the parametric approach and kind of two ways to correct for measurement error within the parametric approach. I'm going to talk now about the non-parametric approach. You know, why is that needed? Because not surprisingly, things fall apart when those two models don't hold. So here's what it looks like. If so these are all the same estimators I just showed you from that parametric framework, but the simulation setting is such that those models don't hold. So the proportion explained is still 0.5, but the models don't hold. So even when there's no error, you see some bias. When there's measurement error and you ignore it, you see even more bias. When you try to correct for that measurement error, you do kind of get back up closer, but still biased. So you can correct for measurement error all you want, but still a problem because the parametric models don't hold. So recent work has suggested a non-parametric approach to define and estimate R. This definition is actually the same as the parametric one I started with if those two models hold, but it's just described a little bit differently. So the treatment effect, I'm going to denote as delta, which is the difference in the primary outcomes between the two treatment groups. And then the residual treatment effect, basically that second term is still the primary outcome in the control group, but then the first term is trying to capture, what the primary outcome would look like in the treatment group if the surrogate marker distribution looked like the control group. And so that delta S is trying to quantify this leftover treatment effect after you account for the treatment effect on S. So kind of forcing the surrogate marker distributions in the two groups to look the same. And so then R is defined as 1 minus delta S over delta. So delta minus delta S is like the chunk of treatment effect that's explained by the surrogate, divided by delta is the proportion of the treatment effect explained by the surrogate. A previously proposed way to estimate this non-parametrically, obviously the treatment effect, you just calculate the average in the two groups and subtract them. For the delta S, the second term is just the average in the control group, and the first term just uses the Nadaraya-Watson estimator of the conditional mean to kind of smooth over S to get that expected Y in the treatment group, assuming that the distribution of the surrogate looks like the control group. So obviously when you don't have S, but you have this mismeasured W, estimation of delta S is affected. Unlike the parametric setup, the attenuation bias here is not in a neat closed form solution. So we can kind of write it out, but it's like in the form of a bunch of integrals. So we don't have a disattenuated estimator here. But I can show you what that bias looks like from our simulation. So here, those parametric models do not hold in this setting, but we're using the non-parametric approach. So when there's no error, things perform nicely. But when there is error, I see some substantial bias. So here, just like in the parametric setup, we're going to use the SIMEX estimator. And it's especially useful here within the non-parametric approach. So for the parametric part, you may have been wondering, like, why do I even need the SIMEX estimator if I have the disattenuated estimator? And you'd be right. You don't really need it because you have a nice closed form disattenuated estimator. But in the non-parametric setup, we don't have that. So this is exactly where something like SIMEX is useful. So it's the same idea. You generate these this additional measurement error. So these more mismeasured W's for iterations, 50 times over this grid of lambda. And then you calculate what that delta S estimate would be for these more mismeasured W's across your lambda grid and extrapolate back to lambda equals -1. And all the same points as in the parametric framework. So showed that consistency and asymptotic normality and derive variance estimates here for for the non-parametric versions of this estimator. So here's what the simulation results look like. So the first two you've already seen. We don't have a disattenuated estimator here, but the last two are the SIMEX estimator using quadratic extrapolation, and then the SIMEX estimator using nonlinear extrapolation, both of them working pretty well. So I'm gonna illustrate these using data from the diabetes prevention program. So this was a randomized clinical trial designed to examine the efficacy of various treatments on the prevention of type 2 diabetes in high-risk adults. We focus on the lifestyle intervention group versus the placebo group. The primary outcome is change in fasting plasma glucose from baseline to 2 years post-randomization, and the potential surrogate marker of interest is the change in hemoglobin A1C from baseline to 2 years. So this is a surrogate marker that is not measured earlier. It's measured at the same time, but is considered less burdensome for patients since they don't have to fast for that hemoglobin A1C. But hemoglobin A1C is known to be measured with error. So the change in fasting plasma glucose from baseline to 2 years was negative 3.3. So lower fasting plasma glucose is better. So negative 3.3 in the lifestyle intervention group and 1.4 in the placebo group. So treatment effect of negative 4.7, that's significant. So here the measurement error variance was not known. And so we had to estimate the error variance. There are, several ways you can estimate that variance. You can use replicates, or you may have external data that you can use. We did have replicate measures of hemoglobin A1C, so we used that to measure the error variance and got an estimate of 0.17. So here are the results. So using the parametric approach, just looking on the left side first, the naive estimate for R was 0.56. And then using the non-parametric approach, it was about 0.4. I'm just going to focus on the nonlinear SIMEX extrapolation estimate. So in the parametric framework, when we use the nonlinear SIMEX estimator, we got an estimate of 0.7, and then about almost 0.5 in the non-parametric approach. So while they both certainly, those estimates increased a little bit when we accounted for measurement error, it's still the case that we, probably would conclude that this is not capturing a substantial proportion of the treatment effect. The confidence interval for both of those have a lower bound that's less than 0.5. So, not really strong evidence that this is capturing enough of the treatment effect. And this is actually a useful conclusion because, some have argued that hemoglobin A1C really could be a good replacement for fasting plasma glucose, like if we could only decrease the measurement error. And what this is showing is that even if you could decrease the measurement error, it doesn't look like it's going to capture that treatment effect. Not that, you shouldn't try to decrease the measurement error for other reasons, but for this specific purpose, it still wouldn't be kind of a good replacement for fasting plasma glucose. So just to a few remarks about this part. So I illustrated some illustrated bias and estimation of R when there is measurement error in the surrogate, and provided methods to correct for this bias in both a parametric and a non-parametric framework. As I mentioned, we showed those asymptotic properties in the variance estimates. And this work is in press at Biometrics, and there's an R package on GitHub called R Surrogate that implements everything described here. So importantly, we have some limitations. So of course, we assume that sigma u squared is known, but we also need these other conditions. So the first three conditions are assumptions that are they're not unique to this work, but are often needed, or some variation of them are often needed in surrogate marker work. And they basically make some assumptions about the monotonicity between S and Y, and protect you against this surrogate paradox situation where you might have the treatment positively affecting the surrogate and the primary outcome, positively associated, and then the treatment negatively affecting the primary outcome, which has happened a lot in practice, and is a very, very bad situation. So these are untestable, strong assumptions that we make. And I know there's work by others, including Mike, looking at how to assess like the sensitivity to violations of these assumptions. And I think that's such an important area of work because, again, there are assumptions that I've had to make across all of my surrogate marker papers and can't really seem to get around them. And much like the work that's looking at sensitivity to like unmeasured confounders. So in all this, in a lot of causal, well, I guess in all causal inference work, you have to assume that you have no unmeasured confounders. And so they have these this approach where you can like simulate an unmeasured confounder that has a certain relationship between your variables of interest and see like how crazy of an unmeasured confounder you would need to just totally reverse your conclusion about whatever it is you're estimating. So if it takes a very dramatic simulated unmeasured confounder to completely reverse all of your findings, then that might make you feel like your results are robust. But if you have an unmeasured confounder that has a very weak association with your variables and that can completely reverse everything, then your findings are probably not very robust. So, I think something along those lines in this area would be really useful. So how much of a violation of these assumptions would you need to be in this paradox situation? I haven't done any work in that, but I think it's a really important area. The fourth assumption there is just needed because I do my kernel smoothing. So I smooth over S in the treatment group and then apply it to the control group. So I need the supports of the surrogate and the two groups to be the same. And also because, go ahead.
So C1, C2, and C3, can't you estimate those quantities from the data and get a sense of whether they're satisfied, or does the measurement error stop you being able to estimate them?
So here I think the measurement error, I think you would need to just basically try to correct for measurement error in doing those assessments. But I have done what you're saying in like other settings where I'm assuming there's no measurement error. And my understanding is that you can get a sense of whether they hold or not, but you can't sort of specifically say whether like these definitely hold. Like in the diabetes example, I can plot these and they do look, it looks like they relatively hold, but I think I can't, I don't think I can come down and say that I 100% think they hold in like all diabetes studies that are going to be looking at fasting plasma glucose and hemoglobin A1C. Okay, what was I can't remember if I said the last thing. So I just need a large sample size because I do the kernel smoothing and the non-parametric approach. So if you have a very small sample size, that non-parametric kernel smoothing is not going to work well. One thing one might ask is whether there's really interest in estimating the proportion of treatment effect that's explained by a surrogate measured without error. So if it were the case that you could never measure the surrogate without error, then do you really care about this proportion for the perfect surrogate, or do you really want to know what the proportion is that's explained by the mismeasured surrogate? So I would argue that if efforts can be made to reduce the measurement error, then I do think it's useful to know what the proportion explained would be of this like perfectly measured surrogate. Because if you can show that the surrogate, the perfectly measured surrogate can explain a lot of the treatment effect and then potentially could be used in a future study, that would kind of incentivize these maybe efforts to be made to reduce the measurement error. Like the diabetes example, sort of the flip side, we showed that actually, even if you did have a perfect surrogate, it's not capturing a large enough proportion of the treatment effect. So for this very narrow setting, perhaps it's not worth the resources in trying to reduce the measurement error there. And then just to mention some of the other work on surrogate markers that I've kind of referenced throughout. So there are certainly methods that we've proposed and others that have proposed to evaluate a surrogate marker. So there are methods available when the outcome is, censored data, when the surrogate marker is not actually a marker, like a biomarker, but is instead an outcome itself that's subject to censoring. There are also methods available to look at how you evaluate multiple surrogate markers. So like in the diabetes example, well, if your outcome is a diabetes diagnosis and you have some potential surrogates like fasting plasma glucose and hemoglobin and all these other measurements that we have, each one on their own might not explain a lot of the treatment effect, but possibly together they might explain a substantial proportion of the treatment effect. You also might have a case where you want to know the proportion of treatment effect that's explained by a longitudinal surrogate marker. So also in the diabetes example, we don't just have fasting plasma glucose or hemoglobin A1C at one time point. We have it like every 6 months. So really, you might want to see if the proportion explained by that trajectory is, more than just what's explained at a particular time point. And then lastly, we've proposed a way to test for the treatment effect using the surrogate marker. So this is, as I mentioned at the very beginning, kind of the promise of surrogate markers. If you can come to an agreement that something is a good surrogate marker, then how do you actually use that to replace the primary outcome in a clinical trial and test for treatment effect? And all of this work has associated software on CRAN or GitHub. And then some topics that I think are really interesting, but I haven't either worked in or finished yet are, so the question of a high dimensional surrogate. So I think this is where I'm interested in it, is this question of can changes in immune activation be captured by changes in gene expression? So for the multiple marker, like multiple surrogate markers method I explained, I mentioned, we basically use like a working model to combine the surrogate markers, and then you have like a composite score, and that's your new surrogate marker. But when you have a high dimensional surrogate, you can't, it's not just as easy as fitting like a working model. So I think that's a really interesting area. I'm also really interested in surrogate markers from wearable data. I know I talked with some of you about that. I haven't done anything with wearable data at all, other than wear a wearable, which I don't think counts. But I think that, it's so fascinating, this relationship between like sleep quality and sleep quantity and like deep sleep and sleep consolidation and heart rate variability and respiratory rate. So all these things that are measured by certain wearables, and the association between all of those things and like cognitive decline and cardiovascular events and strokes. Like, I think that is a potentially really cool area where this research, more globally on surrogate markers could be really useful. And then lastly, I have been working on, and I think this is also interesting, this problem of heterogeneity in the utility of a surrogate marker. So like even in the example I showed today, hemoglobin A1C was explaining like 50% of the treatment effect. But we actually see in the DPP data that differs by certain subgroups. So the proportion explained is different for whites versus blacks versus Hispanics. So I think developing methods to evaluate that and test for that is a really important area to kind of work on in the future, and certainly implications of that heterogeneity for future studies. So if you're using the surrogate to replace a primary outcome in a future study, but you know that the strength of the surrogate varies by patient characteristics, like that seems problematic. And it seems like something you're going to need to carefully consider when you try to use the surrogate in the future. I don't have a solution for that. I just think it's an interesting problem at the moment. And that's it. Thank you. We'll take any questions.
I ask a question? And so you talked about a large sample. So I can think of 3 problems that you might get if you had a smaller sample. 1, you get bias estimates. 2, you'd get, the confidence intervals coverage rates wouldn't be very accurate. Or 3, you might just get high variability of a point estimator. So which, is it all of those that go wrong, or what goes wrong in small samples with your non-parametric method?
I mean, honestly, when I've tried to apply it to much smaller sample sizes, my first place where it breaks is just literally in the estimation part, because the like the bullet before that was basically like, I need the supports in the 2 groups to be the same. So that gets much harder when, I have, let's say I have 20 surrogates in one group and 20 in the other, and I have I do my conditional mean function in one group, and then I come to the other and there's like 3 data points that's outside of the range that was in this other group. I have to do some kind of extrapolation to fix that problem. So just on the very basic level, I basically get like an error in the estimation approach. But even when I do that extrapolation, the biggest problem I tend to see is in the bias. I'll get bias, at least in the few times where I've tried it, but that could have been just my simulation setting. Yeah.
So the coverage rates would be okay at larger samples, would they have confidence intervals?
I mean, there's one paper where I think we did it with like sample sizes of 200, which is smaller. Like the diabetes example had almost 1,000 in each treatment group, and usually I do about 1,000. And at 200, I think our coverage rate started to look not so good, but they weren't that bad, maybe like 92. It wasn't so bad. Well, cool. If there's no other questions, that was easy. I'm happy to, if you have any questions, you can email me. Happy to send a copy. I don't remember if this is online yet, but happy to send a copy if anyone's interested.
I might just ask, while you're here, the error variance, the sigma squared u, if you don't have an estimate of that, are you just out of luck, or is there anything you can do there?
So that's a good question. So I mean, you have to get an estimate from somewhere. When I was working on this, I at first didn't really realize I had replicates of hemoglobin A1C because I've never needed it for any other analysis I've done with this data. So for a while, I was like delving into this online world of there are estimates out there for like the measurement error for hemoglobin A1C from like different, I don't know what it's called, like lab tools. And different companies will like publish different measurement error estimates. But like, I didn't know what lab or what company was used by DPP, so I didn't know which one to choose. And then I realized that I did have replicates, and so I could use that. But what I've also seen people do is consider like a range for the measurement error variance, which I think is a cool idea. So if you have, for us, it was like 0.17, but maybe, if you guessed like it's probably between 0.05 and 0.3 and look across a grid for that, that might be something that at least tells you, what you might expect with less or more measurement error variance. So I don't think you're completely out of luck, but usually there's some way out there to get some reasonable estimate or a range of estimates, I think.
I see. I guess following up on that, I'm just wondering, so if you, instead of wanting to say, here's the proportion of the treatment effect we could explain if we had measured this perfectly, I could imagine in some of those circumstances where the goal is reduce the error variance, you might want to say you have to get the error variance below X in order to explain this much. Is that a straightforward thing to compute from what you've shown?
I mean, I think I haven't thought about that, but I think you could do that if you like. So for me, the measurement error variance was 0.17, right, for hemoglobin A1C. And I'm with this method, I'm basically saying what the proportion explained would be if the measurement error was 0. So if I instead went through everything but assumed, I think with the SIMEX estimator, yeah, it seems like with the SIMEX estimator, you could basically pick.
Just extrapolate to not all the way back to -1.
Exactly. Yeah. Extrapolate back to something, whatever you're, if you think you can only get the measurement error, like you can get it down, but not to 0, maybe just to 0.1. It seems like you should be able to do this whole procedure and just like see then what would your proportion be at that 0.1. Mm-hmm. Yeah, that's a good point.
So, like, ask another point. So, I mean, people were quite critical of apprentice criteria and this sort of proportion explained because they didn't really have a very good causal type of interpretation, right? And
yeah,
you could go outside the range 1 to 0 to 1, and also. So, do you have any comments on that? Does your R ratio sort of overcome that, or is that still just it's a choice of how you want to summarize?
So my R is so those 3 conditions that I have, those 3 assumptions, like ensure that my R is between 0 and 1. But yeah, I mean, I totally acknowledge that there are like limitations with this measure, and people have asked me like, oh, if the proportion explained, if I estimate it and it's like 0.99, then can I use it in a future trial to replace the outcome? And I'm like, no, please don't do that. But I because I think you basically are going to have to like apply all the methods out there and see, if they all basically show that the surrogate marker is a like good surrogate marker, then I think we can be confident that it is. But they all kind of have strengths and weaknesses. And some of the other methods I read about, they'll come up with like 3 different quantities you're supposed to look at. And I mean, I get kind of lost reading it myself because I'm like, well, what if those 3 don't all say something is a good surrogate? Like, what is the conclusion that you make? And like, people aren't using there's still clinical papers coming out today that use just those 2 simple regression models to look at a surrogate marker, which I don't know how that's possible. But yeah, that's partly why I want to have this this surrogate marker workshop in Mexico to because I feel like I don't know the answer to that question. What measure should I be using? If someone can convince me not to use this approach, but use something else, like happy to do it. But as it stands, I feel like they all have their strengths and weaknesses. And this one is just, it is used so widely and it is very intuitive. I don't know if that really answered your question.
Yeah, no, I'm just sort of getting you to talk about what you think about that. Sort of this causal aspect of it.
Yeah.
It's just sort of like, I view it as like an improvement of two regression. It's sort of a variation and improvement on the fitting the two regression models, right? That's sort of,
Yeah. Yeah. Okay. Well, thank you all very much.
Thanks. Thanks,
Bye.
everybody.